Welcome again, friends. In this particular video lecture, we are going to discuss about the meiosis process. In last three videos, we have already talked about the mitosis. If you have not gone through those videos, please go and watch them, and then you can learn about the meiosis, because meiosis is much more complex process than mitosis. So we can divide the meiosis process into these two types: the meiosis 1 and meiosis 2. So what is going to happen in the meiosis? The one parent cell is going to divide into two daughter cells, and those cells will be haploid. So if the parent cell is diploid, in the meiosis 1 itself, the cell is going to reduce its chromosome numbers. So one parent cell which is diploid is going to divide into two daughter cells in the meiosis 1, and then in meiosis 2, these two cells will divide to form four daughter cells. Okay, so this cell has, let's say, the diploid chromosomes. They are diploid. This cell is diploid, and then the two cells which are formed after the meiosis 1 will have the only one set of chromosomes, which means they are haploid. And then let's go into the meiosis 2. These sister chromatids will be separated in these four daughter cells. So ultimately we will have four haploid cells. But remember one thing, the DNA content is less than these chromosomes. So if this cell has 4C chromosomes, 4C DNA content, then these cells will have only 2C DNA content. But the chromosome number is not getting reduced. These cells, if these cells have 23 chromosomes, then the daughter cells will also have 23 chromosomes. So these are the few things you should notice about the meiosis. So let's learn about the meiosis 1 first, then we will move to the meiosis 2. So just like mitosis, meiosis is again divided into two phases: the karyokinesis and the cytokinesis. But let me just write karyokinesis, and second is cytokinesis. Karyokinesis is again divided into four phases: prophase, metaphase, anaphase, and telophase. What is the difference between the karyokinesis of the mitosis and meiosis? This prophase 1, because these are the processes of meiosis 1, then we will call this karyokinesis 1, and prophase 1, metaphase 1, anaphase 1, and telophase 1. But the prophase 1 is much more longer than mitosis prophase, because it is also again divided into 5 subphases. This prophase is again divided into 5 subphases. First is the leptotene, second is zygotene, third one is pachytene, then diplotene, and the last one is dikinesis. So see, the prophase 1 is again divided into 5 subphases: leptotene, zygotene, pachytene, diplotene, and dikinesis. The sequence of these phases you can remember with the mnemonic or lazy paddy. Lazy paddy. So L for leptotene, Z for zygotene, P for pachytene, D for diplotene, and the second D for dikinesis. Okay. So in this way you can remember the sequence of these subphases of the prophase 1. So let's just see what happens in these phases of the prophase 1. In the leptotene, cell will start to condense its chromosome, just which is the beginning of the prophase. So just like the prophase of the mitosis, cell will start to condense and condense its chromosome and to disappear its nuclear membrane. So chromosomes will be condensed and they will came the homologous chromosome will come to close to each other in the leptotene. Then in the zygotine, chromosomes will form a proteinaceous complex. Two homologous chromosomes will form a proteinaceous complex called the synaptonymal complex, and they will just like join to each other in the zygote. So they are forming synaptonymal complex. This process is called as synapsis, and these fibers are called as synaptonymal complex. I hope it is visible to you. Synaptonymal complex. Then in the Pachytene, cell will, these two homologous will do a crossover, just like that. The non-sister chromatids of the homologous chromosomes are crossing with each other. Non-sister chromatids. So these two, which are homologous chromosomes, and the non-sister chromatids, the one chromatid of the first chromosome and the other chromatin of the second chromosome will hug each other, will cross over each other for the process of recombination in the Pachytene. Okay. In the Pachytene phase of the prophase 1, what will happen in the diplotene? And one more thing I have forgot to mention here. In the Pachytene, the recombinase enzyme helps in the process of recombination. So what will happen? The chromosomes, the genes present on the first chromosome will be shared with the other chromosome, and the genes present on the second chromosome will be shared with the first chromosome. So genes are shared with each other. So this process will be done with the help of recombinase enzyme, and the new combinations of the genes will be formed in the Pachytene. Okay, so the process of recombination of what? Recombination of the genes will be take place in the Pachytene. Then let's move to the diplotene. After completion of the recombination process, this synaptonymal complex will be disappeared. What will happen? The synaptonymal complexes disappear. So you will not able to observe or see the synaptonymal complex in the diplotene. But chromosomes are still crossing and having this structure called chiasmata. Okay, so they are not separated yet in the diplotene. So you will see the X-shaped structure called the chiasmata. Chiasmata in the diplotene. And in the dikinesis, after completion of the recombination, the homologous chromosome will be separated, and you will see the recombination of the genes in both the chromosomes. So what is different than mitosis? In the mitosis, only decondensation of the only condensation of chromosome take place in the mitosis prophase, but here the homologous chromosomes are getting are crossing with each other, and they are sharing with their genes to each other, and they are forming the new combinations of the gene, which leads the process of you can see the variations which we see in the organisms if they are doing sexual reproduction. So their gametes are produced by meiosis. So once they are producing their gametes, the genes should be um, forming the recombination. Genes should be recombined to form the new combinations of the genes, because it is very necessary for the process of evolution. So this is the prophase 1 of the meiosis 1. The meiosis 1, the metaphase 1 is much more similar than which is very similar with the metaphase of the mitosis. The metaphase, about the metaphase 1 we will discuss in the next few videos. So I hope it is understood by everyone, the prophase 1 of the karyokinesis 1 of the meiosis 1. So this was about it for today. So we will meet in the next video. Till then, hit the like button, subscribe to my channel, and just share these videos with your friends. Thank you so much. Radhe Radhe.